Mr. McLean. In this video segment, we're going to be taking a look at section 2.3 in the AP Bio course dealing with chemistry of life. And what we're doing is we're picking up with where we left off at in terms of our chemical characteristics associated with atomic structure and position of the electrons. So when we last went over this material, we talked about how the outermost shell of the atomic structure, where the electrons would be at, is referred to as our valence shell. So the electrons that are in that outer valence shell are going to do basically one of three different things. They're either going to be gained from one, from another element, another atom, I should say. They're going to be lost to another atom, or they're going to end up sharing electrons with another atom. So when we take a look at these things here, they're either going to be some type of a transfer or some type of a share. And that interaction, whether it's sharing or transferring, is going to be the building blocks of a chemical bond. So the interactions that are going to hold things together chemically. So when we start looking at the different types of bonds that exist, there are characteristics that are associated with it that are going to give it unique properties. So just by taking a look at what elements are interacting with one another, just by looking at the periodic table and having an ability to identify the valence configuration, you can predict the type of bond that you're going to get. So the first one we're going to look at is called a covalent bond. And you want to think of covalent bonding as sharing. So you're going to have electrons that are going to be shared between two or more atoms in order to complete the outermost valence shell configuration for the elements that we're looking at. So a classic example of a covalent interaction would be a molecule of hydrogen. Okay, so in this particular case right here, we have an atom of hydrogen, and hydrogen has a single electron. So, it wants to fill up that outer shell. And one of the things that we're going to find is that in many instances, when you have only one electron in a valence shell, it's going to tend to lose that. But in the case of hydrogen, the position is so close to the positive nucleus that there's a real strong attraction to that. So it doesn't lose it easily. And then the other thing that we're looking at is you're taking two atoms of hydrogen. So they both have an equal attraction for that electron. So that attraction for the electron is the electron affinity and the electron negativity. So both of these atoms want those electrons. In fact, if you look at this picture, you can kind of see that they're kind of pulling the electrons from each other, and they're kind of hybridizing. So, in this case right here, if I were to click over to my, my drawing pad here, I could probably show you something that would depict kind of what's happening with our hydrogen. So if I said, all right, here is, oh, sorry about that, close that down, click that back over here. So if I go right here and say, all right, well, here is my one hydrogen, and this hydrogen has one valence electron, and we'll change the color up, and here's my other hydrogen right here, and this one also has a single valence electron. This one needs to gain or lose an electron. This one needs to gain or lose an electron, or the other option is that they share. So if we bring these two together, it kind of looks something like this. So you've got a hydrogen, and you've got that single electron right here, and then we have the other hydrogen, and these shells kind of overlap like that. So if we take a look at it, the two electrons that are needed are either here or here. So they're sharing. All right. And because these two elements, or these two atoms of the element hydrogen, are equally attracted, that's going to give us a certain characteristic that we're going to talk about that would be nonpolar. All right. So that's one way we can go through there. Now, when we make a molecule, it's going to have two or more atoms that are held together with covalent bonds. And the number of shares or pairs of electrons that are being shared is going to indicate whether you're dealing with a single, double, or a triple covalent bond. As we start to look at how we would depict the structure versus the molecular characteristics, from a structural standpoint, this would be a molecule of hydrogen, and it's showing that there is a single bond between these two atoms to make up that particular molecule. The molecular formula is just telling me that there's two hydrogens. So the subscript with that, this would be a molecular formula, this would be a structural formula. Like I said, in biology, a lot of these structural formulas become more relevant because we start looking at interactions on how they come together. Excuse me. So as I go over here, we start looking at this setup right here, and we're going to notice that in the case of oxygen, it's not working the way I wanted it to, but in the case of oxygen, we could see this double line here, okay? That indicates that there's a double bond there. So if we go down and I give myself a little bit more room to scroll, here I'll zoom out a little bit and move over. Oh, that's not working so much. All right. But if I go in here and I said, all right, well, I've got oxygen, and we know that an oxygen atom has 8 electrons, so that means that the valence shell is going to have 6. Okay, so I'll draw this one here. I'm going to draw it this way. We got 1, 2, 3, 4, 5, 6. Okay, so I'm just depicting it that way. And then I'll draw another oxygen really close to that. 1, 2, 3, 4, 5, 6. So if you take a look at it, these two from this one can be shared. And likewise, these two from this one can be shared. So when we look at it, what are we doing? We've got 2, 3, 4, 5, 6, 7, 8. We look at the red one, we've got 1, 2, 3, 4, 5, 6, 7, 8. So in this case right here, we have a double bond. Okay? So double bond means that we're sharing 2 pairs of electrons. I'm not getting a good clean right on that, but anytime you have a double bond, you have 2 pairs of electrons being shared. If I jump up here and just give you one other example, we have nitrogen. Okay, so if you go through there, nitrogen is going to have 5 valence electrons. So I'll just draw it this way: 1, 2, 3, 4, 5, and then I'll grab another one. We'll make this one green, and up here, 1, 2, 3, 4, 5. Okay, so if we're sharing each of these right here, let's take a look at it. 2, 4, 6, 7, 8. 2, 4, 6, 7, 8. So nitrogen, triple bond. Okay, so those are some things that we will see absolutely as we go through there and we study some different options on that. So kind of zooming back out of here, we can see things like oxygen and water and methane. Now, our next chapter spends a lot of time focusing in on water and the difference between how like water with its covalent characteristics are different than methane. So I'm going to hold off on that a little bit there. But like I said, the covalent interaction that takes place between those different elements there allow us to build those molecules. Now, one thing that we have to take a look at is how elements really want to grab that electron. Okay, so you have to consider that the elements ideally want to be stable. Not that they have like a personality, but they want to be stable. They want to have their full outer shell configuration, very much like the noble gases. So if they're an element that has 5, 6, or 7 valence electrons, they have a tendency to have a stronger pull for those electrons. If you look at something that has 1, 2, or 3 under most circumstances, they have a lesser um, affinity and electronegativity. So in general, if you're looking at the periodic table, and we wanted to make a general statement as far as where we see the trends going, I guess you could probably say that if you're looking at the periodic table, we find that when you move in this direction, okay, when you move from left to right, you're going to have a increase in electron affinity. And when you move from here to here, so this upper area right here, when you start to look at this particular portion, so you'd have helium above that, and you'd have this last family or of noble gases coming down through here. So what I'm talking about is you're looking at elements such as fluorine and chlorine. They are going to be very, very aggressive at taking electrons. And we're going to talk a little bit about um, some of the properties that they have as a result of that. All right. So electronegativity is how aggressively something's pulling on the electrons. Now, when we have a covalent bond, there's two general categories of covalent bonds. You can have what's considered a non-polar covalent, where the atoms are going to share equally. So if I go back to this picture right here, hydrogen is sharing equally because it's not like one hydrogen is stronger than the other. Same thing is true with oxygen. And we could also say that's true with methane. So, for the most part, we have a very similar pull for those electrons. But when we get something that's polar, polar means that there's going to be a slight difference in charge from one end of the molecule to the next, because of the uneven charge or the uneven attraction. So water is the quintessential example of a polar molecule. Oxygen has a strong pull for electrons, very high electronegativity. Okay, hydrogen, even though it doesn't want to give up its electrons, it will, um, you know, essentially share unevenly with oxygen. Um, the analogy I would say is it's like siblings sharing but not sharing equally. So you can quote unquote share but not share equally. Oxygen holds the electron a little bit longer, so therefore it has a slightly negative charge right here. And then the two positive ends of the hydrogen molecule are because it doesn't hold the electrons quite as much. So we have a whole section where we get into the polarity issue, and I would honestly say that this is one of the concepts that carries through into many different chapters throughout the course. So we'll definitely build on that. All right, so to wrap up some of the other types of bonds, whenever you have an atom that's losing electrons to another atom that's gaining electrons, so in our classic example of sodium chloride, sodium with 1 electron doesn't have much of a chance of holding that. Chlorine, we said, was a very aggressive oxidizer. So in this case right there, the sodium becomes positive and the chlorine becomes negative. These are ions. So a charged particle, positive would be the cation, negative would be the anion. Together they form a ionic bond. And they tend to form these crystalline structures. So when you think about salts and things of that sort. Um, it says most of the strongest bonds in organisms are covalent bonds. The other ones are weaker. Okay, I'm not saying that ionic bonds are weak, but they're weaker than the covalent bonds in this particular setting. We also have hydrogen bonds, which are going to be the strong interaction between the slightly electronegative atoms or molecules interacting with one another. So whether you're talking about water molecules because when you think about water, water interacts with other molecules of water with a hydrogen bond. In this case right here, we have ammonia. So you have this slight negative charge right here, and up here you have hydrogen with a slight positive charge. And the interaction that's taking place right through here is going to form a covalent bond. I'm sorry, a hydrogen bond. Okay, so that hydrogen bond kind of holds things together. We have a couple other types of bonds that are also seen in various interactions and forms of life. One of them is called a van der Waal interaction, and they sometimes talk about hotspots of positive or negative charge. And one of the things that sometimes we can, this image, why this is on here, it's talking about the interaction where a gecko can actually climb on a certain surface vertically, seemingly against gravity. But the idea is that they can create these hotspots where there's separations of charge, and it allows for an interaction that allows them to climb vertically, basically, in these different areas. So we'll talk more about that and see some other examples. So shape and function kind of go hand in hand. It's determined by the position of atoms within the valence orbitals. So one of the things that they might do is they might go in there and form things such as tetrahedrons and various other configurations. And one of the things that really becomes critical for from the standpoint of biological studies is interaction and recognition between cells and various other molecules. So if we take a look at this, the drug morphine, okay, obviously it is capable of having an impact where it's going to block pain and induce some changes at the cellular level. Um, if we take a look at natural endorphins, okay, so you may have heard of, a situation where a person has what's classified as the runner's high, a naturally produced pain reducing sensation. This molecule right here, which is naturally produced, will match up with these endorphin receptors on the surface of the brain cells. Okay. Morphine, which overall doesn't look the same, but the point in that molecule that fits into the receptor is very, very similar. So the point that we're getting to is when we start looking at things like drug interactions and cellular interactions, shape becomes very critical. And if you want to follow back to the chemical interactions, it gives us some idea of how that shape is determined. So hopefully that was helpful. We have one more video in this chapter that will kind of wrap up the content.